So how does DNA replication get started? And I think this is quite a fun little question, but we have, this is a bacterial DNA. In many cases, bacteria will have a closed circle of DNA because their DNA is not too long and you can kind of get away with that. And our DNA, which is much longer, is in chromosomes and it is not circular because it would get tangled and bad things would happen. So, in the case of this circular DNA, there is a thing called an origin of replication. And this is also the case for our DNA, for our straight DNA, non-circular DNA, whatever. There are things called origins of replication. Now, in the circular DNA, there's one origin of replication in the circle, and I'm going to use black because I want the black color. So you have an origin of replication, say it's here. And then what happens is your replication machinery goes around, starting from there, and it creates kind of all new strand. Well, it can just go around the outside, can't it? So it just goes around the outside, or it just goes around one side, and it's just going around and duplicating. Let's just clarify that. So it'll go in both directions from the origin of replication. We'll go in both directions from the origin of replication. So we have our first plasmid, and then we have our plasmid, which started with the origin of replication, and from that point, you kind of get like a bulbous thing happening. Because now here, we've actually got a double strand there, or we've got a double strand forward of the origin of replication, and we've got a double strand backwards of the origin of replication because of the directionality of the DNA. And this is a term that I might as well get out of the way now, but DNA is anti-parallel. And all that means is that the two strands run in opposite direction, of 5' to 3' and 3' to 5'. And we'll talk about what that means in a minute, but if you just store away in your brain that it's anti-parallel, it's parallel but inverted, that's all you need for now. Right, so then what happens in human or non-circular DNA? And also this kind of like, might as well complete it, but it kind of goes all the way around until eventually you're able to butt off that little bit in the center. All right, so now what happens in human DNA? So in human DNA, we start with a straight stretch, and on our straight stretch, we have origins of replication. And like before, the DNA is anti-parallel, which means that it replicates in different directions. And so we get to our origin of replication and we start replicating this way. And then we have another one before which we were actually going like this. We go up to the third one and we're after it and we're before it. And we go around. And I'm wrong. That's not how it looks. This is a learning experience for me as well. So we kind of have a bit of this, little bumps. And it gets replicated. Now, in these little gaps, you have your double-stranded DNA now, and it's going in both directions in here. And so eventually those guys are going to meet up, and you'll have a complete double-stranded DNA thing, and it'll be complete. And so that's how DNA kind of gets replicated in humans and bacteria. Now we'll go through some of the machinery next.